Okay, some of you guys were asking about gene regulation, and there's a lot of different terms here: positive gene regulation, negative gene regulation, and how they all work out, and then also negative and positive feedback, which makes it a little bit confusing because they're the same terms. So, kind of a quick summary for you guys. First of all, gene regulation can be either negative or positive. So, when we talk about negative gene regulation, by definition, the thing that you guys should be comfortable with is that negative gene regulation is gene regulation in which a repressor protein is used to actively stop transcription at any point in time during that gene regulation. Usually it stops transcription by blocking polymerase from doing things. Positive gene regulation is any time that a chemical actually helps polymerase do transcription. So negative gene regulation blocks it, positive helps it. Okay. Within negative gene regulation, there can be either negative feedback or positive feedback, which they've renamed repressible and inducible. I wish they didn't do that, but that's what they did. Negative feedback means that when the chemical is present, it stops transcription. And positive feedback means that when the chemical is present, that it allows transcription to occur. So, and I know that we just talked about how negative gene regulation is something that stops transcription at any point in time. Let's take a look at some examples of negative gene regulation and see if we can straighten that out. So if we take a look at the, let me think here, the TRP operon. This one here, even though the little website says lac operon, this is the TRP operon. This has been studied in bacteria, and this is how it works in bacteria. Pretty similar in eukaryotes, but for the general audience here, this will have to make sense. So this is the TRP operon. Tryptophan is a amino acid that your body needs. And if it doesn't have it available, you have a gene, a set of genes, gene E, D, C, B, and A here, that are transcribed by polymerase. And polymerase transcribes those 5 genes into 5 different proteins, which then combine together through different foldings. And then what they do is they synthesize tryptophan. They make tryptophan for you. So when you don't have tryptophan, your body makes it for you. Typically, there's a regulatory gene someplace else here. Now remember, you can be negative regulation or positive gene regulation. And it involves a repressor. So this regulatory gene by itself transcribes this mRNA, which becomes this protein, this repressor. When there's no tryptophan present, this repressor is inactive. It doesn't really do anything, which makes you wonder, like, well, why would you make it? Because when you do get tryptophan, your body doesn't need to spend the energy to make these proteins and then synthesize tryptophan. So what your body does, or what bacteria do very efficiently, is that when tryptophan becomes present, then what it does is it binds into this little active site on the repressor protein, changing it to active, and then that repressor protein goes into the operator region here, and it blocks polymerase from going down the track here to transcribe anything. So by definition, remember that negative gene regulation is any time that you use a repressor protein to block transcription, that would be your negative gene regulator. Okay. Now the question then becomes, is this positive feedback or negative feedback? So this is negative gene regulation. Is it negative or positive feedback? Keep in mind, negative feedback means that when the chemical is present, it stops transcription. And this one is when the chemical is present, it allows transcription. In this case, the chemical in question is tryptophan. This is why they're called the tryptophan operon or TRP operon. When this chemical is present, it looks like we stop transcription. So by definition, this would be a negative feedback or a repressible enzyme or a repressible gene. Okay, so that's that example. Very similarly is the lac operon. And because they look so similar, you guys try to kind of make them the same, and they're not exactly the same. Similar principles. Lactose operon. When you eat lactose, you need an enzyme called lactase, and lactase helps you break down lactose. When you don't have lactose available, you don't want to make that enzyme. It's a waste of time. So what ends up happening here is that there is this lactose operon up here. Here's the regulatory gene. Regulatory gene transcribes mRNA, which becomes this repressor protein. But in this case, this repressor protein is active right away. And what it's going to do is it's going to block transcription of the lactose operon, these genes. Well, why would it stop transcription? Well, because you don't have lactose in your system, and you don't need these genes to make lactase for you to break down lactose. So it's just a light switch and it says stop. Polymerase cannot go any further. When you get lactose into your system, so here's lactose. When you get lactose into your system, it goes into the active sites of the repressor. The repressor is inactivated then, and it cannot block polymerase. And polymerase is allowed to transcribe as needed. It makes these three enzymes, which combine together to form lactase, which then break down all of the lactose available. When all the lactose is broken down, then we're right back to this scenario up here, where the repressor protein is active and stops transcription. So going back to our flow chart here, is this negative or positive gene regulation? Well, at any point in time, did something block transcription from occurring? And the answer is yes, and it was a repressor. So that falls under our category of negative gene regulation. Then the question becomes, is it negative feedback or positive feedback? Remember, chemical present, it stops transcription. Chemical present, it allows transcription. Take a look. When the chemical in this question becomes present, lactose, when lactose becomes present, transcription is occurring. So therefore, this would be a positive feedback. This would be an inducible operon of a negative gene regulator. Okay, and then our final example here is positive gene regulation. So again, when we get away from regulators, then we start talking about positive gene regulation. Remember, the definition of that was any chemical that helps transcription occur. And there's a number of examples of them, most of which are positive feedback systems for you. So I think we'll just kind of take a look at what the book had. Cyclic AMP, adenosine monophosphate, that is basically a stripped down version of ATP. And the cyclic AMP stuff interacts with other chemicals in your body called CAP, and it's an enzyme, and it activates the CAP. And again, this is in prokaryotes, but it also happens in humans as well. So this activated CAP then goes to a CAP binding site, and it pushes the polymerase along and allows transcription to occur. So this would be your positive example. When this positive gene regulation example, excuse me, when this chemical is no longer present, then the CAP is inactivated down here, and it doesn't bind with polymerase, and it can't find the promoter. So polymerase needs the CAP's help. Okay. Notice that in this question, there's a repressor that's listed over here, but it's not really doing anything. Ultimately, the question comes down to, is there something helping transcription occur, or at least polymerase get pushed along, or is there anything blocking transcription? There's certainly not anything blocking transcription. There is certainly something helping transcription occur. So that's going to be our positive gene regulation. And for our purposes, they're almost always going to be positive. I shouldn't say almost always. For our purposes, they are going to be positive. So inducible. This chemical is going to show up and it's going to help transcription occur. So there you have it. There's the ultimate idea of gene regulation and how you guys should understand it at its roots. Hope that helped.